indicating where we are in relation to the stage. So what we do here is we go low mag and scan an overview, and then use that overview to do subsequent scanning. So I go live. I'm going to only use the green channel for the overview map. Don't need more than one for a map. It's really bright. I'm going to turn it down. Okay, and then I'm going to navigate around. If it was 1.25, this would be a lot easier. I'm going to zoom all the way out. I'm still on resonance scanner. Right, so I know the sample ends about there. I want to make a register a marker. And I'm pretty sure that one should be somewhere around here. Too far. Okay, register. Okay, so I know the sample's in between here. Now I'm going to register an area like so. These are the two navigation points that I used in this position list, which I'm just going to get rid of. So now I've got basically a stitched area using 10x.
Okay, so basically what you're doing is, this is the entire size of my tissue, at least what that I want to look at.
Yes.
And do this Z scanning thing for each of these squares inside the big square.
That's correct. Now every time you draw a registered position or position one, some of these are drawing tools, some of these just press a button. So this just registers the position where you click. This one registers a certain number of columns and rows based on the values you put here. This is one that you draw. This is a polygon that also that you draw, like so.
Okay.
So each time you do this, it takes all of the settings that you currently have set. It could be the objective, it could be the scan area, it could be the detector, it could be these things, and it allocates it to that registered area.
Okay.
So you need to be careful of that. If you ever make some changes, which I did, you need to update those changes to the registered area, otherwise you're going to be potentially using the wrong settings. So delete that. So this one,
so ideally do all the settings and then do the
yeah, so one thing I had on was I was using frame averaging. I don't care about averaging for this map, it's just a map. So I turned that off, and now I need to highlight the registered area either by clicking this line or clicking on the box and say update registered acquisition parameters, and that's it. And so I'm ready to apply this map now. And start.
So it's building from the bottom, like the first squares. And
yeah, if we were on a 1.25x or a 2x, we'd probably be done by now.
Okay.
Which is why it's so advantageous to have a low mag.
Yeah, right.
You're going to be doing this, I assume a lot of people are going to be doing maps and then doing other scans. Because what this gives you the ability to do is just do multiple different things programmed across the map. You can change, so in this corner I can do 20x Z stack 2 channel here, 40x M8CL like a stitch area Z stack, also incorporating time 5 channel thing. I can do something else here with a different zoom and so on. You can just do whatever you want, and then you press go, and it'll just go and do that whatever you said.
Okay, you can, like, if it says it takes an hour, you can just walk away.
You just walk away, yeah, and let it go and do its thing.
So what is this staining for this sample?
I assume it's GFP.
GFP.
So to start with, if you have a tissue, you would probably want to have a stain that stains the outline of the cell, and one that has a nucleus, and then whatever you're interested in. kind of
That
as
would-
a 3.
that'd be
That's kind
useful.
of
Again, it depends on the question you're asking, but yeah.
but like as a starting point.
Yeah, I think it would be useful.
Yeah.
Yeah. So talking about the pinhole size, the pinhole aperture, you can actually change the size of the pinhole here by using this slider or putting in a value. Auto means it's going to be one airy disk unit, which is a calculation based off the objective. You can change, you can have it tighter or wider. So if you want it to be more wide field, you can zoom it out, but that means you're going to get more out of focus light. It's going to be less confocal. Sometimes you want that. Sometimes you want more depth of focus rather than optical sectioning. And we'll switch over to, let's use the oil objective. So we'll switch over to 60. Let's go live. And now we're doing sectioning.
So having this map is actually really useful.
Yeah. Alright. So now I'll set up each channel again. Activate these. We're not worried about the transmission channels. So red is very bright. I'm going to turn it down. See what happens if we change this collection window of the server. That changes quite dramatically. Okay, let's check the high-low. Saturation looks pretty good. Just going up and down, seeing if there's any bright spots. And it's fine, then we're going to go to the phase 2. Check this one. So this one, I don't think there was much. It's sort of like not as continuous as label, whatever it was. So it might not even be anything here in this particular field. I'm actually going up and down right now. I'm not seeing anything. Alright, so maybe we save some time and not forget about this phase altogether for now. And we'll go back, skip this off. Phase 1. So live again. No. Okay, so we'll go to the MATL tool by hitting that. And we'll register a smaller area here, 2x2, press OK. Okay. And so we're incorporating within this a Z stack, which we didn't have selected before. We do now, so we'll hide that and say update parameters. And it's taking 30 minutes for this acquisition. So we'll change this to something like that. We might change this speed resolution down by half, and we'll again update acquisition parameters. And now we're getting 3 minutes, which is acceptable. And start. a lot cleaner. Post, you know.
Yeah.
There, open up this image. Okay, so here's our image. We need to stitch it together, actually. So we highlight the thing that we want to stitch. We press the stitch button. So currently these are just neighbouring each other. They're not actually stitched into one final image.
Okay.
It shows you a preview. Make sure smoothing and matching algorithms are on.
Mm-hmm.
Press execute. Sometimes it can take a long time.
Because if you have a lot of things that go together, right?
Mm-hmm. Sometimes you have to walk away and let them come back if it's big enough. Okay, so that's done. Now, it's not going to automatically close this window. You're going to close it or press cancel. And close. And now we've got the image. We can do the volume view again. Again with the volume, it's recommend you go into the viewer, use the volume settings. Check here. And take the wire cube off. Background is black. Okay, and then let's do that. Put a bigger image on the screen. Yeah, and so still probably should take out some of the red. So we're done. With that table. Channel 2. Hold down shift to zoom. Okay, that's better. What's that? Okay, there you go.
So this time we have a shallower sample because we told it not to
Only,
do
yeah.
too much.
Also, we disobeyed the sampling recommendation because, you know, there's we reduced it from like, I don't know, 44 to 15 slices. That means the step size, there's going to be gaps. Yeah, so, and you can kind of see, if you look in this green, you can kind of see the gaps, you know. It's not as continuous as it could be, but yeah, you get the idea. I mean, it's for demonstration purposes, you understand what's going on. Yeah, and you can create a movie from here pretty easily as well. So you can go to movie, keyframe, you can give it certain rotation values. You can actually just move it. If you hold down shift, you have this ability to zoom like this. And so you can do things like just keyframe your own organic positions. So like add keyframe, and then we can do rotation, keyframe.
So keyframes are like...
They're like log points. Yeah, they're like log points within the movement. So here we've got this angular rotation here, X and Y, so it'll spin like this. How many divisions, what angle, you know, you can specify that, and these are sort of the more rigid way to do it. You can also, most people just use this, but you can also just, you know, I can get this and spin it, keyframe, and exactly, I can, you know, and then I might want to focus here, I might want to go like this, and I want to move here, and you know, you can just create whatever you want.
Is it the image or is it the location? So is it just so that you can go
exactly
back
what
to
you're
it?
seeing here? It's kind of like when you press keyframe, it's being logged, and then it will extrapolate the movement in between. So if I export this, change this to 30 frames per second. So see, it's doing what it's doing that spin that I did.
Yeah, okay.
And now that's saved as a movie file.
Okay, yeah. Oh, that's cool.
Yeah.
And it can also save some sort of universal image file.
Oh, there's heaps of ways you can play with this. So I've got like, here's a 3-side view, for example, so I can, you know, this is the Z view. So as I go through Z here, you know, I've got the single view. I can scrub through Z like this. I can go tile view. I can export each individual tile. each individual Z position. If you go file, export, there's lots of ways I can do this. So different file types I can export as a bunch of TIFF stacks, all in the series or only some in the series, all the channels or only some.
And this stuff is the stuff that you said the freeware can also do.
I don't know what the freeware is able to export as. The freeware is able to, because this is an OIR file, which is a proprietary file.
Yeah.
You can't just open it up in a Windows image viewer. You have to use a special microscope imaging software in order to be able to do things like navigate through Z, understand that connects with that and that connects with that, and so on.
Yeah.
And so the image J with the Olympus image viewer or Evident image viewer will open this native file and give you the ability to do these sorts of things.
Yeah.
I don't know to what extent it will allow you to export the file like I just went in here. This gives you all options that you could possibly want, different file types and things like that. You can even export as a TIFF stack and import that in some other third-party software to do something with some analysis or whatever.
We can also put the scale bar.
Yeah, you can put the scale bar, and how you do it and where you do it and what software you do it completely changes as well. This is pretty rudimentary scale bar, but we've got CellSense. This is another software, which is an additional license that does so much. Not only can you just add annotations to scale bars and lots of things like this, like drawing objects or whatever, but it does a whole bunch of analysis. So, and it does it very simply. Like you just get an eyedropper tool and start painting what it is you want to measure. Everything on the screen that's like that, could be like the color, like the shape, like the channel, like the whatever, starts to get highlighted. Then you start gating on what it is you want to measure. So you can say, okay, everything over this size, exclude. Everything that's this shape, morphology, whatever, shape factor, exclude or include or whatever. Then you end up with a really tight measurement of whatever it is. And then what it's measuring is, you can almost, anything you can possibly imagine, like the, not just the intensity or the size or how much of this, if something is red is also blue, but heaps of stuff.
When we should put the oil drop?
When?
Yeah.
When you, that objective is not going to work without oil. So as soon as you want to use the objective, you want to put the oil on there. And then when you're finished, make sure you clean the oil off with ethanol ether 70-30%. And don't use Kim wipes, use lens cleaning tissue. I've got lens cleaning wipes with me, which I use to clean, but you can get those. They're basically a 80% alcohol solution with a fabric-y sort of I don't know what it is, some kind of cotton type material.
Like those ones that you can get
like what you use to get for glasses. Yeah. I actually have used, what they give you that you might be concerned with this is when you're imaging really thick, you're going to get less light delivered the deeper you go and less collection the deeper you go. So one of the things we have here is called Bright Z. We've got a tool window, I go to Bright Z. We have the Bright Z tools here. So as I'm going through Z, so let's say we turn this on during manual, you can turn it on during your Z series, during your Z stack. But the way it works is like this. Your laser, your voltage, your gain and your offset can be adjusted as you go through Z. So let's just say, I'll register this as a position here. Right? So at this position, at this Z position, we're using this laser intensity, this HV. Let's say when we go deeper, we're going to increase and increase and increase and increase. And we say register. And now see it's changed. I'm going to go deeper again through Z. I'm going to register these increased values and register. And you see we're starting to create a curve.
For the actual position.
Yeah, and you see here, I'm actually moving the Z. And look, these levels are changing now because it's following this path.
Curve, yeah.
So this is something you can use to help you when you're imaging through Z. Only if you're struggling to deliver light deeper. It's just one technique you can use to improve the Z imaging. Okay. It's going to be useful if you've got really thick, really dense. You can't get light through.
Yeah, I guess basically if the normal Z doesn't work, it's the troubleshooting.
Yeah.
Yeah. Okay.
There's another set of things which I can show you, but it's not. I'm not going to bother because it's going to take too long, and no one's going to use it. I think these are the synchronization and sequence tools, which, like I said, if you're doing stimulation type experiments, but I don't think I was doing that. So, and it's going to take too long to go through it. It is detailed in this manual.
Okay.
But yeah, what I've covered here is, pretty much
The stuff.
80%, 90% of what people are going to do, I reckon.
Great.
So it's a good place to start.
Yeah, I feel like I had enough input.